can everyone hear me okay? Yes. So hi, I'm Belle. I'm a PhD student at the University of Southampton, and I am investigating the prospects of label-free microscopy for research and diagnosis of osteosarcoma. Sure, that doesn't make too much sense right now, but hopefully it will at the end of this talk, which I'm super thankful to be giving today. So just very, very brief, as most people in the room will already know this, osteosarcoma is the most common primary cancer of the bone. About 160 people will be diagnosed every year in the UK, and of these, it's predominantly children and young adults. It usually starts in areas of rapid bone growth, such as around the knee, and the current approach for diagnosis is a very lengthy, multimodal approach that can really delay the diagnosis of the disease, which highlights a really urgent need for early diagnostic markers of the disease, as well as new methods that will allow early, accurate diagnosis of the disease, and tools that will enable accurate removal of the tumor so no cancer cells are left over. Which leads me on to label-free microscopy, which is sort of my main focus, and I will be focusing on these two processes known as second harmonic generation imaging and two-photon excited fluorescence. So essentially, to describe it really simply, label-free microscopy techniques involve directing a really intense laser beam onto a completely label-free sample. And why that's quite special is usually if you want to look at any kind of biological sample, you need to add a fluorescent dye or a tag or a stain to make sure you can see that, whereas this is completely label-free in nature, as it's exploiting the physical sort of properties of the laser. And when the light is re-emitted, it can be picked up by a detector, and you get these really, really nice images. Second harmonic generation in particular is very useful as it specifically images certain structures, and a biological molecule of interest that it images really well is collagen. And two-photon excited fluorescence can image the sort of autofluorescence of the sample, and you can overlay the two like so to see the collagen interface within tissues. These approaches have started to be explored for sort of examining and diagnosing a number of cancers. And so just to give a bit more background, collagen in cancer. Collagen is very important as it's the most abundant protein in the human body, and it provides structure and support for cells. Many, many diseases, including cancer, will exhibit really abnormal collagen as one of their sort of symptoms. In breast cancer, it has been shown that an increased amount of collagen is associated with increased probability of developing breast cancer, as well as increased aggressiveness of that cancer. However, a lack of therapeutic tools and diagnostic tools to actually look at this, the abnormal collagen within cancer, can really sort of contribute to the poor prognosis. I just wanted to put this up here. This was some work done back in 2006 now by another group who cultured breast cancer cells within the lab in a collagen matrix, and they imaged it using SHG microscopy, the same techniques that I use. And what they actually saw, they saw a previously sort of concealed ability for the tumor cells to actively remodel and reshape the collagen to form almost like a protective barrier around the tumor, and that can, do things like protect against the host immunity or like chemotherapy. And what was even more frightening was that at invasive margins of the tumor, so at different areas of the tumor where the cells were starting to migrate away from the primary site, they actually modeled the collagen completely differently, almost into tracks away from the primary tumor, and they could actually use that collagen to migrate. So this SHG imaging uncovered this really scary sort of ability of cancer cells, and yet no one's really used these techniques to look at primary bone cancers. And so my project is asking whether these kind of microscopy techniques can be used to examine and diagnose osteosarcoma. So for my hypothesis and aims, or what we predict, is that osteosarcoma collagen signatures discovered using these kind of microscopes will allow distinction from healthy bone and may predict the metastatic ability of the cancer. So in short, collagen will be a very useful biomarker of osteosarcoma. And how I want to do this is characterize human bone and osteosarcoma tissue using these label-free microscopy techniques and investigate whether there are unique collagen parameters associated with the cancer. So the method I use is I investigate human bone and human osteosarcoma biopsies, image them with these fancy laser microscopes. I've optimized it to enable imaging of sort of the full biopsies that I have. And then what I do with this is run it under some fiber analysis so it can extract all the information there about the collagen, and then I can run some stats and see whether there are any differences there. And essentially, can these label-free microscopy techniques be used to characterize human bone and osteosarcoma? The answer is yes. So this is a, like I said, completely label-free image of human bone. The collagen is shown in green, and it's in these really sort of clear circular structures around the blood vessels that run through bones. So bones are very highly vascularized structure, and these circular sort of structures are very, very well known to be in bone, and you see these little pits are where the bone cells live, and this is the blood vessel that runs through the central canal. It's all very well organized and all very neat. If you then come to a human osteosarcoma biopsy, it's completely different. It's completely lost all of that organization. You have the sudden changes in the collagen matrix that you're seeing here, and things like the vasculature is a complete mess. You don't have the nice organization that you have in normal bone. So this is really obvious by eye to me that both the normal bone and the osteosarcoma looks very different, but what we really need is to extract, quantitative data from that, numbers from that could be actually used to diagnose between healthy bone and osteosarcoma. So my aim was to get this diagnostic signature of osteosarcoma based on the collagen that I'm seeing with these new techniques, to see if we could use these techniques in the clinic for diagnosis. So what I found when I run these SXG images under CT-Fire, which is the software that I use to extract all the information, was that human osteosarcoma has significantly shorter collagen fibers than normal bone. And that's in males and female comparisons, so I kept the two separate. And as you can see, this is sort of the whole distribution of collagen lengths that I'm getting from each of the biopsies, and you get much, much longer collagen fibers in the healthy bone and much, much shorter ones in the osteosarcoma bone. And that's both for when you look at the average length of all the fibers there and the average length of the longest fibers. In both male and females, you have a much, much lower collagen fiber length in the cancerous tissue. And just to, why that's really exciting is that proves that there is a difference that can be picked out using these microscopy techniques, and that could allow for diagnosis between the tissue types. So just to summarize, label-free microscopy has demonstrated promising potential as a really highly sensitive tool for researching osteosarcoma and diagnosing the cancer by looking at the collagen matrix. Collagen is abnormally altered in osteosarcoma, which demonstrates its potential as a biomarker of the disease as well as a therapeutic target. And collagen, it's specifically the collagen fiber length measurements that really allowed me to discriminate between the healthy bone and the osteosarcoma tissue, which again highlights the potential for cancer diagnosis. Now I wanted to really end on a sort of, why does this matter? What could it really allow in the clinic and in terms of real outcomes? So the success of near real-time automated diagnosis during surgery has been shown in brain cancer patients using label-free microscopy techniques. So this is another label-free microscopy technique. It's not the same one that I use, however, the principles are the same. And this group here, Holland et al., just last year did a huge clinical study with almost 300 patients. And what they had was one of these label-free microscopes at the bedside after the brain tumor biopsies had been taken. And in conjunction with artificial intelligence, sort of deep learning methods, they could diagnose the cancer in under 150 seconds. So literally as soon as a biopsy was taken, it could be diagnosed. And why that's so special is, with current methods, if you have a pathologist in the hospital that can diagnose it for you, they could probably do it in about half an hour. But realistically, many centers don't have the pathologist there. The samples will be sent off. There's then a huge extra delay in the diagnosis, whereas this could literally do it at the bedside when the biopsy was taken to a really, really high accuracy. So this just kind of, really validates the fact that if we can find a diagnostic signature of osteosarcoma using these techniques, then maybe we could really provide proof that this could be used in the clinic to diagnose these really, horrible cancers early. With that, I'd like to thank my project supervisors and everyone in my lab group for helping me. My charity that funds my project is Hannah's Wilbury Wonder Pony, who are an amazing charity, and I think they're going to be working closer with the BRCT as well. And thank you all very much for listening and having me here today.
So have you been able to look at pre-osteosarcoma bone? Because you're looking at tissue that's already osteosarcoma and demonstrating nicely that you can differentiate that from normal bone. But actually what we really need is a tool to identify the bone before it really becomes osteosarcoma and is truly damaged. So that's my first question. And then my second, more speculative question is, could you imagine in the future that we might be able to develop the technology to do this without actually having to do a biopsy? Because then you could screen people if they were coming with bone pain to see if it was actually osteosarcoma without actually starting to chop into the leg.
Yeah, for sure. Really great questions. For the first one, the data that I showed today is just stage 2, stage 2B osteosarcoma, and I actually have human tissue right down from stage 1A all the way up to stage 4B. So now I'm looking into, does this technique also have the sensitivity to, discriminate between the stages as well? Coming on to sort of precancerous tissue, I don't have access to human tissue for that, but I've received tissue from Professor Aggie in London, who's got a mouse model of osteosarcoma, and he's sent me tissue of both sort of tumour tissue as well as bone tissue with a very, very, very early cancerous lesion before it's actually developed into a tumour. And that's another thing that I'm currently imaging, and I'm going to see whether I can detect really early collagen changes before the full-blown tumour is there. Regarding the second question about whether we'll be able to get it to a point where we don't need to take the biopsy, that is the kind of 50-year plan. There's a lot of people, I'm a really interdisciplinary project. One of my supervisors is a bone biologist and my other supervisor is a chemist, and I use all of his techniques, and I work with many engineers and physicists and chemists, and there are fellow PhD students now working on making these deep tissue imaging. So as of right now, they're very you need a very thin section of tissue, but now they're trying to manipulate these techniques to be able to, detect deeper into the tissue. And so the idea really is the hope that in, 50 years' time or whatever, you could go in, scan your arm, not taking anything out with these laser techniques, and then they could diagnose a cancer or whatever in that way without having to take the biopsy. But that's other people's expertise, and they're trying to develop these techniques. So for me, it was more just proof of concept. Is there something different in the osteosarcoma? Can we see it? Can I prove that it's there? And yeah.
Right. I just think from the surgical specimens you get from the surgeons that you collaborate with, often the piece of material bone that's taken out, there's non-involved bone, so maybe you could get some of that to test that. But yeah, let's hope it doesn't take 50 years to get the scanning work. Good luck with that.
Thank you very much. Thank you. Any other questions?
That was really interesting. I had two questions. One was the second one that Sue asked, but the other one is, bone tumors are very heterogeneous, and I don't know about this, but obviously you must do. Is this sort of disorganized collagen on the shorter fibers, whatever, everywhere in the tumor, or is it going to depend on where you actually take the sample? Or could you? What I'm trying to say is that if you took different samples from different parts of the tumor, would you see the same type of disorganized collagen and smaller fibers?
So I think, as I showed in that in the earlier slide with the group's work, it will depend on where from the tumor you take the sample, depending on. However, I did a lot of optimization at the beginning of my imaging when I was deciding, how wide a field of view of tissue do I really need to investigate to get that representative, and I started with quite small, well, not even quite small, like 300 microns, which is quite common in the field, and then I did it right up to the over a mil, which is the size of the biopsies. And what I found is for human bone and osteosarcoma, I needed around 700 microns or more to really get a representative image. Otherwise, you are, like you said, you're picking up, collagen from a really small area of the cancer, and it often threw up results that were completely untrue. Like there was, and I went back and repeated it on when I actually looked back. If I'd used a smaller field of view size that some people had recommended at the beginning, it said there was no difference, in the collagen between human bone and osteosarcoma. So I was really glad that I took that time to really find how big a field of view do I need to go. Obviously, there are limitations when you're working with human tissue. I work with what I get, but yeah, so I have taken that into account when I'm doing my experiments.
Thank you.
Thank you.
Thank you. Excuse my ignorance. Can you clarify for me, when you talk about disorganized collagen, do you just mean disorganized, or are you referring to an excess of collagen?
So it will really depend on the cancer. What from my sort of investigations that I've done, the actual amount itself doesn't seem to change that much, but the actual collagen is formed in fibers. So you have molecules of collagen, and then they form fibrils, and then they form fibers. And the actual fibers themselves are much, much shorter in the osteosarcoma. As you can see, you lose. It could also be in part in healthy bone. You have very organized blood vessels that you see in these central canals, and in tumors that's completely gone. It's very well known that the vasculature within tumors is a complete mess, and so it might be that to do with that, the collagen's not forming these same sort of organized structures. So from what I've found, that really the length of the fibers has been the most telling between cancer and healthy bone, but I'm also looking to try and analyze it in other ways to see if I can pull out things like, you know, in bone, it's quite obvious that there's these circular structures. Maybe I can try and pull out differences in the actual shape of the collagen matrix as well.
So there'd be no link between something like Ehlers-Danlos syndrome, for example, where you've got an overproduction of collagen?
You would have that as well, obviously. With normal bone going through life, you have a very sort of neat balance between the formation of bone and the degrading of bone. So all through our lives, our bones are constantly being changed, dependent on things like exercise. Often in cancers, you'll have a complete shift of that. Some cancers, you'll find that the bone is really degraded, so you'll have a loss of collagen. Other cancers, you'll have that the bone, you know, is really upregulated, and there'll be big bony masses which have a lot of collagen in there. Again, I've also got chondrosarcoma samples and giant cell osteosarcoma samples, so again, that'll be interesting to me to see between bone cancer types, is there a difference in the collagen that I'm seeing? So yeah, that's very early stuff. But
Thank you.
Very, very quickly, sorry. And I might not word this right, but is it going to be limited still to the same sort of biopsy issues that there are at the moment, with a small biopsy not being able to have a cancer cell in it to be diagnosed? And I think this is what Sue was getting at, is what the limitation is it? I can't word it right. You were saying that you needed a bigger sample.
yes.
But
Bigger
if you're not getting a bigger sample in a biopsy?
Bigger sample, but definitely a biopsy is more than enough to be able to diagnose with what I'm doing for sure. So I'm talking microns rather than millimeters. So as long, it's not even ideal to really have to take that biopsy, because we know that that leads to other problems like metastatic migration using the biopsy trapped and everything. So eventually we want to get to a point where we don't have to take the biopsies at all, which is where, like I said, the engineers come in and they're trying to develop these tools to be able to go deep tissue. As of right now, however, if you can, you know, diagnose a biopsy in like 2 minutes, instead of have to send it off to another center, you know, how many more patients could you see in a day? So I think there is, even though it's still at the biopsy level right now, there's still huge benefits of using, you know, a different tool. So.
Right, thank you.
Thank you
very much.